Good morning, everyone. We are going to start the session. Welcome to this session. And well, we're going to start right away. So Dr. Margarito Amaral will give us a brief introduction and some overview in the field.
Thank you so much, Gianni. It's my pleasure to be sharing this with you. And welcome to everybody to this workshop. The big question, and what we are also trying to address here in this workshop, is whether CFTR is just an anion channel. So there is evidence that actually CFTR also plays other roles in the cell, namely in what we call epithelial differentiation, cell differentiation, and regeneration. Okay, it's not just because I tell you, because in God we trust, everybody else should bring data. So what's the data that's supporting this? Several lines of research already for some while have documented that there is impaired delay or delayed epithelial differentiation in CF epithelium after injury versus non-CF epithelium. There are also several reports saying that there is increased cell proliferation. There is loss of cytoskeleton organization in CF cells and significant defects in tight junctions and barrier function impairment, which we all know is really important. More recently, there have been reports with data showing that there is abnormal wound healing. And if you think about the CF epithelium and all the challenges that it is subjected to, we may easily understand why once it has been challenged by bacteria, by particles, whatever, it becomes a problem in terms of healing that epithelium. So altogether, this data indicate that CF epithelia are not as differentiated / polarized, because tight junctions and epithelial barrier are, of course, key points in the polarization. So in other words, it's not as specialized as non-CF epithelia. So I'm now going to show you some data from my own lab. And we show here in native human bronchial tissue that we get from explanted lungs that this marker of proliferation, Ki67, it's almost absent in non-CF bronchi, but it's quite active in CF tissue. In primary human bronchial epithelial cells, there is enhanced proliferation of the CF cells, and this we have seen with different genotypes as shown here, and these are the control non-CF cells. This is recapitulated in cell lines like the CF bronchial epithelial cell lines, where we also observed upregulation of this proliferation marker in the CF cells versus non-CF. So altogether, CF native tissues, primary cells, and human bronchial cell lines have enhanced proliferation. That's the increased number of Ki67 positive cells. And as shown here, there is an increase in the expression levels of Ki67. So what about regeneration? As I just mentioned in the beginning, some years ago, it was proposed by the group of Edith Puchel, who some of you might remember, that there are 4 steps after the epithelium has been injured. So the first step, the cells, and these are, of course, the basal cells, which have this multipotency to differentiate into other cells, they start migrating, then they proliferate and in step 2, and then in step 3 and 4, they start differentiating into the different cell types of the epithelium. And this is the, here what I'm showing is airway epithelium. And in step 5, there is complete regeneration of the epithelium that was injured. And of course, there is mucociliary clearance and all the properties. Now, what happens to the CF epithelium when it is injured by some challenge? It goes quite well up to here. Here in step 2, I already showed you that there is increased proliferation, so that's not a big issue. But it's in these steps, the latest steps of differentiation, that there is a problem, at least a delay in getting to the final stages. So because it's not properly differentiated, this creates opportunities to additional damage. And of course, one of the most important is the door, let's put it that way, that it's open for Pseudomonas. So Inés Panconian, a postdoc in my lab, has developed this high throughput, I'm not showing it here at high throughput, but it's a high throughput wound healing assay. So what we can see here, excuse me, is that there is a wound, and then with time, in time-lapse imaging, we can see that the wound is closed nicely after 24 hours. So for the CF cells, approximately the same wound, if anything a little smaller than that one, it's still not closed after 24 hours. So we can actually put in a graph the rate of closure, and this is done automatically, and we can see that the CF cells actually have delayed closure, because after 24 hours, there's still some area which is not covered by cells. Now, this appears like a paradox, because I told you that the cells differentiate faster, but in terms of closing the wound, they're actually slower. So, yeah, this is just to show that the same difference is also recapitulated in CF cell lines. We have a hypothesis for why this is happening, because I'm not going to show you today, but we do have data showing that the actin cytoskeleton is very disorganized in the CF cells. So these cells, in contrast to the wild type ones, actually miss direction. So they're not proliferating and moving by whatever attracts the non-CF cells to each other to rapidly close the wound. Yeah. Another hypothesis that we put to try to explain what is happening here with CF cells is this process of epithelial mesenchymal transition. Is it occurring in CF? So there are actually 3 types of epithelial mesenchymal transition, which, as you can imagine, is exactly the opposite of epithelial differentiation from mesenchymal into epithelial. So in type 1, epithelial cells transform to motile mesenchymal cells. These are the ones that, for instance, in cancer are mostly responsible for metastases. And this happens in physiological, normal situation, which is embryo development. So these cells are still mobile. Type 2 of EMT, or epithelial mesenchymal transition, is in wound healing and organ fibrosis. So as I've shown you, when there is a challenge or an injury to the epithelium, the cells, the mesenchymal cells, are able to sort of close this wound, and they do this by becoming more primitive, in other words, being mesenchymal, or the basal cells actually become with a mesenchymal phenotype. In type 3, it takes place in cancer cells, so this is no longer physiological, so causing tumorigenesis is far from being a physiological normal situation. And as I said, they imply cancer progression and metastasis. So these are the three types we know of EMT. And now I'm going to provide some evidence that actually EMT, all three types occur in CF. Because we have found years ago in the transcriptional signature of CF that there is quite a cluster of genes which are known to be involved in EMT. There is impaired differentiation, regeneration, as I just shown you, and there is also compromised development. And we know, and even yesterday we heard about trachea malformations, so there are issues in the airway branching, and of course CBAVD, or either bilateral or unilateral absence of vas deferens. And there is, this is, you know, the emerging challenge, increased risk of cancer. And of course, as most people with CF now getting the modulators and living longer, which is amazing and fantastic, but the reverse of this coin is that they do have increased risk of cancer. And in some cancers like colorectal, it's about 5 to 7 times higher than the normal population. So there is some evidence from what I've shown here that there is this EMT occurring in CF cells. Okay, so again, this PhD student, Margarida Quaresma, she looked at bronchial tissue. This is native human tissue, again from explanted lungs. And she looked at some epithelial markers. Of course, CFTR, beta-catenin, zoonin, so the protein in tight junctions, and E-cadherin. And of course, CFTR is not there because this is CF epithelium, but the other markers, it's not that they are downregulated. In some cases, they are even upregulated, okay? However, the mesenchymal markers are strongly upregulated, some of them like vimentin or collagen 1. So altogether, what this shows is that mesenchymal markers are upregulated, epithelial markers are disorganized, because if we zoom into this tissue, we can see that the cytoskeleton is heavily disorganized. So this is what we call partial EMT, so the cells are not completely going into mesenchymal state, but they are not fully differentiated epithelial cells either. So making a very long story short, Margarida actually looked at several transcriptional factors which are causing and have been related to EMT. One of them is YAP1, which is in the Hippo pathway. And what she found is that in the CF cells, here delta F cells, the YAP1 has a more internuclear, sorry, it's not intracellular, it's internuclear localization than in non-CF cells. So if YAP1 is more in the nucleus, here it's correct, it is more active. And that could explain why we observe this mesenchymal or partial mesenchymal phenotype in F508del cells, in general in CF cells versus non-CF cells. The big question is, can we restore F508del by restoring the differentiation status of the cells? Because if the cells have this big handicap of not being fully differentiated, it's certain that they will not have the machinery responsible, for instance, for trafficking, which should be in place in a fully differentiated cell. So by knocking down some key transcriptional factors that cause EMT, what Margarida was able to do and to show is that, so I'm showing here YAP, but also TED4, TWIST1, and CEBP beta. So all these factors actually contribute when we knock them down, so reducing the mesenchymal state of the cells, we can actually rescue some of F508del, not just localization, but also the function as shown here by this electrophysiology patch clamp data. So knocking down these transcriptional factors partially rescues the traffic to the plasma membrane of F508del processing and function. Now the opposite question, by rescuing F508del or any other CFTR variant, can we restore the differentiation status of the cells? And here, this is a sort of complex slide, but I will try to lead you through it. What we can see here very clearly is that there is, versus the black bar, which is the control, we can see that in the presence of CFTR modulators, which we know rescue F508, so ETI, we can see that the mesenchymal markers actually go significantly down. They're not yet at the wild type levels, but they are reduced under ETI. So the EMT modulators actually partially restore the epithelial phenotype. Okay, what about if we knock down one of the key transcriptional factors I mentioned, YAP1, together with the modulators? So what we can see here is that with knocking down by siRNA of YAP1, this is actually additive to the effect of the modulators. So this sort of tells us that there is still scope to improve the action of CFTR modulators. So in summary of what I've shown you, CF is an epithelial differentiation disorder, and we are not used to looking at it like this. Proliferation is enhanced, however, regeneration is impaired. EMT, so epithelial mesenchymal transition, is active in native CF tissues, primary cells, and cell lines. And the transcriptional factors responsible for EMT are actually consistently upregulated in CF. So the rescue of CFTR, of mutant CFTR by current modulators, only partially reverts EMT and partially restores differentiation, demonstrating that functional CFTR directly maintains the epithelial status, so in non-people without CF, of course, and prevents EMT. And this might be the reason why some authors have called CFTR an anti-tumorigenic gene. So, and this happens even without any infection or inflammation. So the bottom line is that there is still room to further improve the rescue of mutant CFTR by modulators, namely regarding the CFTR role in maintaining the epithelial, I'm sorry, you cannot see it, but in maintaining the epithelial differentiation status. Okay, open questions, and I hope that we will have some discussion after the talks of the presenters here today. Which progenitor cells require CFTR expression and function to differentiate properly? Into which cell types do these progenitors differentiate so that actually those that require functional CFTR? Which progenitor cells should we target to rescue CFTR, for instance, by messenger RNA therapy, gene editing, and so on, so as to restore the physiological function of the CF airway epithelium and, of course, the other epithelia? And in which cells should the basic defect be restored to the ultimate benefit of people with CF? So I'm going very quickly through the summary of our talks here today. First, we have William Shaheen, I hope I'm saying your name correctly. William is going to talk about GMEC cells, which are glandular myoepithelial cells, and explaining that LEF1 is a transcriptional factor that enhances the regenerative capacity of these cells, which is something we look forward to because, as I said, this is impaired in CF. Interestingly, these transcriptional factors is enriched in ionocytes. And William's hypothesis is that LEF1 overexpression can promote ionocyte differentiation by SMG myoepithelial stem cells. The second talk by Amirala Nazari, I hope I'm saying it correctly again. So pulmonary ionocytes are expressed in the mucous tubules, you have here the ionocyte, and ciliated ducts of these submucosal gland cells. Normal mucin viscoelastic properties require bicarbonate secretion by ionocytes, as also shown by several authors. So the hypothesis here is that ionocytes' function in the submucosal glands is to produce bicarbonate secretion. We look forward to your talk. The third talk by Ruth-Love Damoa, actually, I should tell your other name because you recently changed your name, but you will show it, I'm sure. She focused on Notch. So we know that Notch signaling is very important in terms of stem cells activation, but also at some later stages after injury. So prior studies have shown that ionocyte frequency is modulated by Notch signaling in non-lung tissues. Notch activation inhibits rather than promotes the emergence of FOXI1 positive cells. And the hypothesis here is that Notch signaling may be relevant for ionocyte precursor differentiation. I'm disclosing already a little bit the conclusion, but yeah, you will hear more about it. And our final talk today by Saira Hamad is focusing on an inhibitor, an antagonist of the ORI1 channel. So it mediates the calcium, ORI1 mediates the calcium influx and drives pro-inflammatory response. So by using this compound that binds to ORI, it inhibits ORI1 activity. So with this, it is expected, and I'm creating some suspense for Sahira's presentation, it is expected that this inhibitor will reduce airway infection, will reduce neutrophilic inflammation, normalize mucus production, reverses lung damage. So, without further ado, these are the 4 presentations that we will listen to this morning. Thank you.
I have a comment, but also, a question. So the primary cells, when you're looking at proliferation coming out of a tissue, I mean, in vivo, there's a lot of injuries. So the replication in vivo and the samples in the lung tissues is probably a reflection of the damage that's occurring. But when those cells come out of that and they go in tissue culture, they're going to remember those signals of, probably even epigenetically. So,
but also cell lines.
Yeah. Oh, and the cell lines, that's interesting too. The other 2 comments are that CFTR can physically associate at the membrane with channels like, or factors like P10 and affect AKT. It can affect Wnt signaling, which is a positive signal for a stem cell.
Yeah.
What are your thoughts about that? And when you think about the mutations that you evaluate in culture, do like homozygous nonsense mutations actually have more significant change in their proliferation or ability to migrate?
Yeah, that's a very good question. Actually, we did show that even variants that cause residual function, they actually cause this EMT-like or partial EMT that I have shown. So the other question regarding the P10, I think that's a very, very important point, because P10 is very actively maintaining the apical versus the basolateral. And some years ago, it was actually shown that one of the first things that Pseudomonas do to the cells is actually they make P10 to be internalized so that the apical membrane becomes basolateral-like. So in addition to all these issues, there is even, you know, this particular effect of Pseudomonas, and maybe this could explain why there is this high affinity for this particular bacterial species.
Yeah. What was interesting about the paper by Edith Puchel on the presence or absence, I mean, it was the physical presence or absence of CFTR. It wasn't its channel activity, and to keep P10 and inhibit P10 at the membrane, that I found fascinating.
Yeah, that's actually very interesting, and that's why we are now recapitulating these studies with G551D. So we do have a cell line which was developed in Ron Crystal's lab, which is a basal cell line, and it differentiates, so it's a non-CF, and it differentiates into various cell types. So based on this cell line, we have gene edited into delta F, but also G551D. So I keep you posted.
Stay tuned.
Hi, Margarita. Jed from the CFF. So generally, I think of CF as being a small airway disease, and the predominant isolation source for the HBs is the large bronchial epithelium. Have you looked in the small airway, and do you see a more significant phenotype where the small airway versus the...
We haven't. That's a very good point, because usually when we do the primaries from the explanted lungs, you know, it takes some time, is great work burden. So we just focus on the big airways, because dissecting the small airways is
the return on investment.
But now I think we should start looking into the small airways. So thank you for the suggestion. Yeah.
Hi, very nice talk, Emanuela Brucia from Yale University. So my question for you is, you discussed a disorganization cytoskeleton, right? And so definitely could be because CFTR is a macro complex at the plasma membrane, but do you think it's only about that, or there is a problem with signaling that leads to cytoskeleton disorganizations? What is your thought in?
We really don't know. That's one of the key questions that we are trying to address. But several groups have actually reported, like John Anaran, I don't think he's here, but reported this issue with tight junctions and, you know, tightness of the epithelium. So what you say is a very valid hypothesis that the CFTR complex with all these direct and indirect interactions is helping the cell to maintain the fully differentiated status. And then this comes again to John's point about whether it's the presence of CFTR or the function that is needed for that.
And just a comment, in terms of disorganizations of the cytoskeleton, I don't think that is just an epithelium issues. We see exactly same issues on macrophage when they get activated, and we see them in macrophage that are knockout for the CFTR. So it may be much more also related to signaling and altering the differentiations of other cells. Right?
Yeah.
So.
And this reminded me of a recent piece of data, still preliminary, that we have with the G551D cell line. It still has some of the defects of delta F, but it's intermediate in the wild type. So both things, function and correct localization, may be required.
Thank you.
Thank you, Margarita, for the beautiful introduction. So I think we can start with the first speaker.
I think we can go.
So Wema is going to talk about the left one and ionocyte.
Good morning, everyone. I'd like to thank Margarita for the great introduction. She already put my research in the context of current paradigm of airway regeneration. That's great. And also, I'd like to thank both chairs for the opportunity to present my currently favorite project today. So, as Margarita mentioned, I'm talking today about LIF1 and how it alters the ionocyte differentiation from several progenitors in the submucosal glands. There are no relationships to disclose related to this presentation. I have asked the gentleman here in the room to dim the lights a little bit, so everyone would get like a chance to resume his sleeping. But all right, how did we get in there? So basically, we have myoepithelial cells of the airway submucosal glands that are known to be reservoir stem cells for some epithelial cell types populating submucosal gland and also the surface airway epithelium. However, it's not known if it specifies to ionocytes or not. At the same time, lymphoid enhancer binding factor 1 is an HMG transcription factor that enhances the regenerative capacity of glandular myoepithelial cells. Interestingly, lymphoid enhancer binding factor 1 is also enriched in ionocytes. Additionally, it has been noticed that both ionocytes and submucosal glands share the same anatomical distribution across species. While you can find them mainly in the proximal trachea of mouse, you can find them across the cartilaginous airways of other species like ferrets and pigs. All of the above led us to hypothesize that probably LIF1 overexpression will promote ionocyte differentiation by glandular myoepithelial stem cells. To tackle this question, we went ahead and used the alpha SMA CRE-ERT2 driver, and within epithelial surface airway, within the airway epithelium, this transgenic mouse model will only express CRE in myoepithelial cells. So we mated these mice with either ROSA 26 tomato GFP, and with CRE recombination, it will excise tomato and stop codon and express GFP, or we made them with a conditional knock-in mouse model. In this conditional knock-in mouse model, CRE recombination will excise the EGFP and stop codon and allow for LIF1 overexpression. All right, so let's treat them with tamoxifen in vivo and see how it looks like. So in wild-type mice or ROSA TG mice, only myoepithelial cells will be GFP positive, while in the LIF1 knock-in or conditional knock-in, myoepithelial cells will be LIF1 overexpressing but colorless. With that, we will prepare glandular progenitors, and then FACS sort them for wild-type myoepithelial cells, which will be GFP positive, and LIF1 overexpressing GFP negative myoepithelial cells on the other hand. This is a long introduction, but I wanted to make sure that it is well known that myoepithelial cells will be LIF1 overexpressing and GFP negative, or on the other hand, they will be GFP positive but wild-type. All right, let's look for ionocyte differentiation or ionocyte expression in these cultures, in ALI cultures. So after allowing them to differentiate and polarize in ALI cultures, we have seen more BSND in red and also FOXI1 in white in the LIF1 overexpressing myoepithelial cells. And this was not only in one CRE driver. We have another CRE driver that's again specific for myoepithelial cells within the airway epithelium, and it's a smooth muscle myosin heavy chain CRE-ERT2. When we quantified that, there was a significant increase, or the LIF1 overexpressing myoepithelial cells had shown more or differentiated more into ionocytes, FOXI1 positive, BSND negative, or BSND positive, FOXI1 negative, or double positives. All of them were higher in the LIF1 overexpressing in myoepithelial cells in both mouse models. All right, how does this, we want to make sure that this is real. It's not just a hoax. So we went ahead and did qPCR to make sure that this is the case. And with qPCR, we have seen an increase. Yes, it's there. In both mouse models, we have an increase in BSND, of upregulation of BSND, foci ion expression, in addition also to CFTR. All right, we have more ionocytes in these cultures. How does this affect the short circuit current? When we run short circuit current, there was a significant increase in the cyclic AMP-inducible GLH101 inhibitable CFTR-dependent current. And this was in both LIF1 overexpressing both Cree drivers compared to the wild type. And this was significantly different. And the same thing applies to the bicarbonate short circuit current here. And we use symmetric chloride and bicarbonate short circuit current here. All right, that was all in vitro. What about in vivo? So we again used the same mouse model, treated with tamoxifen, and we are looking now in tracheas for GFP positive wild type myoepithelial cells and their progeny that hopefully include ionocytes, and also the same thing for GFP negative LIF1 overexpressing myoepithelial cells. 35 days later, we sacrificed the mice, and we did a whole mount staining for BSND as an ionocyte marker. And let me orient you a little bit to this thick Z-stack. It's about 200 Z levels, and it goes all through the thickness of surface airway epithelium as well as the submucosal glands. That's why I'm showing two XY views, one at the surface airway epithelial cells level and also at the submucosal cell level. As you can see here in magenta, BSND, we have significantly more BSND positive cells on the surface as well as in the submucosal gland of LIF1 overexpression in myoepithelial cells. And the wild type side, it was hard for me to find any single 63X view that has both ionocytes in both surface airway epithelium as well as the submucosal glands. When we quantified that, either total number of BSND positive cells, we found a significant increase in LIF1 overexpression in myoepithelial cells, both on the surface as well as in the submucosal glands. And when we looked for myoepithelial cell-derived or lineage-traced BSND positive cells, again, it is still significant. When we looked for the other cell types that are not myoepithelial cell lineage, they are not significantly different. They are the same. All right, so far I have shown that LIF1 promotes ionocyte differentiation by glandular myoepithelial cells, both in vitro and in vivo, and this was associated with an enhanced CFTR-dependent short-circuit current in ALI cultures. This means that, and also the GMAC lineage accounts for most of the increase in airway ionocytes after LIF1 overexpression, and thus I think GMACs are good targets for cell-based therapy to correct CF and COPD phenotypes. All right, now there is a question. What about the other non-myoepithelial cell types in the submucosal glands? We have different cell types. We have serous, we have mucus, we have some other progenitors. Do they contribute to the LIF1-induced ionocyte specification? To tackle this question, we went ahead and used the same mouse model again. We are using alpha SMA Cree that's mated to conditional knock-in mouse model, and then we treated in vivo with tamoxifen. The infect sorted for GFP negative LIF1 overexpressing glandular myoepithelial cells to separate them from GFP positive non-glandular myoepithelial cells or wild type myoepithelial cells. And then we worked further on the GFP positive non-glandular myoepithelial cells. We either treated them with TET-Cree, and this will induce Cree recombination in these cells and make them again GFP negative, LIF1 overexpressing, but again, these are non-myoepithelial cells. Or we leave them with vehicle, which will make them controls GFP positive non-myoepithelial cells. Then again, we differentiated them or polarized an ALI culture, and then stained for, again, BSND and CFTR this time. So here again for another orientation, this is another Z-stack for ALI culture of a whole mount stained ALI membrane. And I'm showing two is the XY views here because I want to show CFTR on the apical domain of the cell, as well as BSND and hopefully secreted globin 1A1 and the cytosol of these cells and the other cytosolic domain view. All right. Interestingly and unexpectedly, we have seen less ionocytes in the LIF1 overexpressing non-myoepithelial cells compared to the wild-type ones. And we are seeing here a lot of CFTR expressing cells that are not ionocytes actually, and these happen to be secreted globin 1A1 positive or serous cells. When we quantified that, we found a significant decrease in BSND positive cells and also CFTR, BSND double positive cells and non-myoepithelial cells when overexpressing LIF1. All right, that's interesting. How will this affect the short circuit current or chloride conductance in these cells? We did the same thing again, and we ran short circuit current, and again we show it correlated with what we saw of ionocyte reduction. There is a significant decrease in the cyclic AMB inducible GlyH101 inhibitable CFTR dependent current in the LIF1 overexpressing non-myoepithelial cells, and this was quantified and came to be significantly different. All right, so these are non-myoepithelial cells in total. What about secretory cells? We know that secretory cells or serous cells express secreted globin 1A1. And at the same time, ionocytes express low levels of secreted globin 1A1. So we thought there might be some lineage relationship between these two cells. So we tried to see what about the contribution of the secretory cells to LIF1-induced ionocyte specification. To tackle this question, we got this mouse model that have a Cree driver with the secreted globin 1A1 promoter. And this means that only serous cells will be expressing or have Cree and Cree combination when crossed to LIF1 overexpression. We prepared submucosal gland cells and her progenitors, and then we have differentiated them on ALIs to polarize them, and then treated ALIs with either 4-hydroxytamoxifen to induce Cree combination and overexpressed LIF1 only, and secretory cells, which will be GFP negative, LIF1 overexpressing, or vehicle to leave them as controls. And then we get these whole mounts stained for first LIF1 to show that some cells will have LIF1 overexpression, and it's here depicted in orange. And you can see we have a bunch of cells that are overexpressing LIF1 and the LIF1 in the 4-hydroxytamoxifen treated group, but there's nothing actually in the control group. For BSND, there was not significant difference in the number of cells that are BSND positive, not significant increase or decrease in ionocyte differentiation between both controls, and it's depicted here in red. And the p-value is 0.2152. All right, so it didn't affect the differentiation of ionocytes. What about the short circuit current? The same thing, so it was not different between both groups. So with that, so far, for my conclusions, we have shown that LIF1 enhances ionocyte specification and anion conductance by glandular myoepithelial cells in ALI. And also it inhibits ionocyte differentiation by non-glandular myoepithelial cells, the other cell types in the gland and the submucosal glands, and it does not promote ionocyte specification by secretory cells. We think that induction of ionocyte specification by LIF1 overexpression in myoepithelial cells will help restore the airway surface fluid and mucus balance in CF patients on CFTR modulators. With that, I'd like to go for future directions, which might be like, which type of GMACs is actually giving rise to more ionocytes with LIF1 overexpression? And what are the intermediate cell types that goes all the way? Because it looks like non-myoepithelial cells don't give rise to ionocytes with LIF1. So it has to be some other undiscovered intermediate cell type, or it might be differentiating directly from myoepithelial cells all the way to ionocytes. And also we'd like to dissect what are the pathways affected, and also we want to see LIF1 overexpression in other animal models. With that, I'd like to thank everyone, every current and previous lab members of the John Englehart lab. And I'd like to thank John personally for his support, and also Jevahir and Kyler for their hard work to help me with this project. Also, I'd like to thank all the members of Yemen's lab and CF Foundation for support. And thank you all for this early morning attendance on Saturday. And I'll be happy to take any questions. Thank you. Yeah, that's a very great question. So for the first part of the question, we have shown that our previous paper by Tom Lynch and other members of the lab have shown that after injury, myoepithelial cells actually in vivo can differentiate to all different cell types on the surface and also on the submucosal gland, and it's almost normal distribution of the cells. It was very functional airway epithelium. For the second part of the question, how does LEF1 overexpression affect the distribution of other cell types? Here's the point. Since ionocytes are rare cell types, they are less than 1% of the cells. I don't think if you augment that to be like 3-5%, it will affect other cell type differentiation. I think they will be mostly, we didn't do the experiment actually to look for other cell types specifically and how they are affected, but the airway surface is very functional, physiologic, and also the ALIs act normally.
Very cool. Thank you very much.
Thanks for the question. That's a great one. I like it.
Hi, thank you for the talk. I'm Kwajanowski from University of Saskatchewan. I was wondering if you could describe the phenotypes that the submucosal glands exhibit when you have LEF1 overexpression in those mice. Did the submucosal glands produce different mucus? The submucosal, the animals alive, they have a phenotype?
So let me, so the question is about how LEF1 overexpression alters the phenotype of submucosal glands. So the mice are alive.
Yeah.
And they are doing fine, and they regenerate all the cell types in the submucosal gland. They are all labeled in non-color or colorless cells coming from myoepithelial cells, and they are totally fine. They are doing well, and I don't think we didn't actually go specifically to look at the phenotype, but speculating, I don't think there's a difference in these cells.
So the viscosity of the mucus doesn't change?
No, I don't think so. The mice didn't show any phenotype. They were absolutely fine.
And the second question is, would you present the data as percentage of cells, so BSND label percent? How do you measure the percentage? Did you count all the cells, or it was a surface area? How do you count the
we count all the cells in the membrane. We do a whole mount stain. We count all the cells and divide them by the total cell number in the membrane, unless we specifically mention that this is a percentage of the labeled cells or lineage traced cells. This is a different thing.
Thank you so much.
Yeah, no problem. Thanks for the question.
Great talk, William. Tom Lynch,
Tom.
University of Nationwide Children's Hospital. Also biased towards supporting William here and his work, but lately I've been rereading some of those papers by Elaine Fuchs on LEF1 expression in hair buds, and she demonstrated using like LEF1 chip and all this stuff that LEF1 can bind to different genomes, be transcriptionally active or repressive. And I'm wondering if you have or plan to look at actual transcriptional changes that occur with the overexpression of LEF1, if that's different in myoepithelial cells versus
go ahead.
Versus other cell types.
So the question is how LEF1 as a transcription factor will affect the gene expression of other genes and other cell types like myoepithelial cells, other submucosal gland cells. And it's, yes, I'd like to do that. It's sometimes challenging because, there are not many options to study that. One of them is painful, like CHEP. The other one is somehow painful again, like single-cell RNA sequencing. And yes, we would like to do so.
Suppose you could do qPCR of certain left one target genes. I mean,
Yeah, yes, definitely we can. But I think the more direct answer to your question is to chip.
yeah, but I mean, who wants to do that, right?
Yeah. Thanks, Tom.
Just a short question. Are there any small molecules activating LF1?
Yeah, that's a good question. We always have been thinking of doing small molecule screen to get something that will do the same, and it will be even easier than applying genes or expressing genes in the myoepithelial cells. Great question. I like it.
Thank you very much, Wim.
Thanks, everyone.
So our next speaker is Amirala Ostad Nazari, and the title is Ionocytes localized in airway submucosal gland mucus tubule and ciliated ducts contribute to mucin unpacking.
All right. Well, as Margarita mentioned, my work revolves around pulmonary ionocytes in the submucosal glands and how they may potentially contribute to mucin unpacking. Firstly, I want to extend my gratitude and appreciation for the invitation to present our work. There are no relationships to disclose. So I think it's fair to say that pulmonary ionocytes are a big topic that is being studied in the field of CF since their discovery in 2018. Many studies have been done to understand what the function of these cells are and how they potentially could contribute to pathophysiology of CF. One proposed idea is shown by Dr. Engelhardt's group, and that ionocytes could be involved in secretion and absorption of fluid, while another study has been done showing ionocyte absorption only of fluid. And our work at University of Saskatchewan has shown with our latest publication that ionocytes could be involved in pH regulation in human bronchial epithelial cells through a CFTR linked with anion exchanger to transporters. And what potentially could be a contributor to fluid secretion and absorption is through the club cells, as shown by other studies. Now, all this really shows is that there's a lot of controversy in the field, still a lot of unknowns to discover and really conclude what the function of these cells are and how and what truly they do in the pathophysiology of CF. So in the proximal airway, submucosal glands produce most of the fluid. I just want to familiarize you with the anatomy of the submucosal glands. The fluid being produced in the serous acini cells gets secreted into the mucous tubule up to the collecting duct and into the lumen of the surface epithelium through the ciliated duct, as the dark regions of this submucosal gland pointing towards the most prominent regions of the CFTR. We know that in CF, submucosal glands are abnormal in terms of their morphology, the fluid that is secreted within them. And recently it was found that actually ionocytes are identified within submucosal glands. We can see that they were identified in the duct and the surface epithelium as well. There's been studies that have been shown in the past that actually bicarbonate and a proper functioning CFTR channel are required for the proper mucin secretion into the airway. Now, when this doesn't happen, what we get is an impairment in mucus detachment, abnormal fluid matrix that is secreted, rheological properties of this mucin are abnormal in the airway. So what we hypothesize is that within the submucosal glands, the ionocytes located within actually secrete bicarbonate that could facilitate this mucin viscoelastic properties. How we're going to test this is through understanding where they are actually located anatomically first in the proximal airway, and then move on into actually analyze their ion transport properties. And we want to do this in native swine tissue. So what we first did, we did a whole mount tissue staining. This is a confocal image on the left. On the surface epithelium tissue, we got a phalidine marking the cell membranes and give us a visualization of what the tissue appears like so, and BSND and FOXN1 marking and labeling ionocytes. MUC5AC marking mucin, in the tissue as well. So the main point from this picture is we can identify opening of the submucosal gland with the MUC5AC labeling the mucin, and we can identify ionocytes within the surface epithelium labeled by BSND. Onto the right, similar preparation, confocal image of the surface epithelium. We can again identify the opening of the submucosal gland with the blue markings of MUC5AC and abundant amount of ionocytes labeling the surface epithelium. So what we did next was completed paraffin embedded tissue staining. We can actually see in these sliced images that BSND again is located on the surface epithelium, and we can see that opening of the submucosal gland in more of a sliced image to get a better visualization, and you can actually see the submucosal gland deeper into the tissue, shown by the serous acinis. On the panel to the right, we were able to identify ionocyte within the ciliated duct as well, as shown earlier, and also ionocytes within the submucosal gland. Now, they're shown to be very minute. There's approximately 1 to 3 ionocytes per submucosal gland, but we were able to identify that and verify that. So we try to look deeper into the submucosal gland. We can see this is a confocal image of a Z stack, and we can see on the left side the arrow pointing towards the serous acini cells, the right arrow pointing towards the mucous tubule, and ionocytes labeled by the BSND and the FOXI1 labeling the nuclei. Panel B and C are the 3D construct of the Z stack, and we can have a better visualization of that ionocyte internally inside the submucosal gland rather than the serous acinis on the side. This image also portrays the same conclusion, better visualization of the mucous tubule and serous acini around the region of this, and then the ionocyte labeled by BSND and FOXI1. Again, panel B and C, 3D construct of that. So what we want to do afterwards is to actually understand what their ion transfer properties are. So we use a technique that we adopted from our latest publication as well. It's called self-referencing ion selective microelectrode technique. But the first challenge really is to dissect these submucosal glands, dissect all the connective tissue that is covering everything there, and be able to label live ionocytes in live tissue so we can conduct our electrophysiology. So what we do is we use a mitochondria dye, and the reason we do that, we take advantage of the fact that ionocytes are rich in mitochondria. So this mitotracker will help us screen for ionocytes. In this left panel, we can see ionocyte labeled by this mitotracker. We identify them, complete our electrophysiology, and then we actually verify that that same screening is an ionocyte with a BSND. And that's the crucial part, that what we measured is actually a function of an ionocyte and not other cells that have high rich mitochondria. So in terms of the actual technique, this panel on the left will help me kind of demo this. So this is the airway surface epithelia. Any high ion transporting cell will create this unsteered layer, in this case sodium, transporting from the luminal side to the basolateral side. Due to this transport, you will always have a lower concentration of sodium closer to the cell than further away. So what we do is that we use an ion selective electrode to measure this difference in ion concentration. In a steady state, we convert this data to the diffusion rates, see how fast they're moving, and identify the ion transport of these cells. So this graph on the right will give you a visualization of where that microelectrode is placed and how we can measure that. So this data, again, is from those verified ionocytes within the submucosal gland. On the y-axis, we have the proton flux over time. And what we can see is that before stimulation with fluoroscoring IBMX, we have a acidification of the luminal side, and after stimulation, we have the alkalization of the luminal side. This suggests that bicarbonate is secreted into the airway through these channels, and it's very consistent with the HBE cells that we saw, and we also see these in the distal airway as well. So the next step was to do these cells actually, or does the bicarbonate that is secreted from these cells actually have any effect on the mucin at all? So what we did was we wanted to inhibit ionocytes first. So we used the IDS to inhibit the ionocytes. And again, as we've shown, we know that CFTR and ion exchanger 2 work together to drive bicarbonate secretion. And the IDS will be a blocker of this anion exchanger. And more importantly, it has no effect on other cells like club cells, and it's not toxic. So we know specifically that we're inhibiting ionocytes. So the technique we used was, it's called FRAP, fluorescence recovery after photobleaching. What we do is extract mucus from the proximal airway. We stain it with FITC dye. We photobleach with a powerful laser a region of this dye, and then it's shown that actually the recovery of this photobleach is correlated to the viscosity of the mucin. So this graph is fluorescence over time. We can see after photobleaching the recovery between the two conditions. And again, this is very preliminary data, but it's showing that when we do block ionocytes, the recovery half-time, which from that photobleaching, is twice as much as the control, meaning that some sort of property of this mucin is abnormal, causing it to be more viscous than in control conditions. So the working model that we're working towards is that in a normal condition, ionocytes secrete bicarbonate into the lumen of the submucosal gland, allowing for normal properties and viscous of the mucin to be secreted. And then in a CF-like condition where we have the mutation of this of these transporters, less bicarbonate is secreted, and which causes increase in viscosity into the airway. Again, as we showed that it was pulmonary ionocytes are expressed in the mucous tubules and the ciliated duct, and as well as the surface epithelium, and these properties that are secreted into the airway actually require the bicarbonate secretion to be normal and proper. And in the future, we hope to locate other transporters and see the other effects of other cells into the mucin. It's important to verify this function as just an ionocyte and not a combination of other things. Measure live pH of the secreted fluid to understand if the pH actually does change in different conditions, and extend this to human excised tissue samples to understand if these results stay consistent in the human airway as well. With that, I want to acknowledge my lab from University of Saskatchewan, especially Dr. Yanowski, supervisor, and Dr. Luan for all the immense help, and of course, the agencies that fund this research and help us to understand more about CF. I can answer any questions. Morning.
Good morning. Great talk. Ian Thornell, University of Iowa. I was wondering if you looked at some of the basolateral transporters that might either extrude protons out of the cell or bring bicarbonate into the cell, specifically comparing those from the ionocytes in the gland to the ionocytes that are on the surface, and whether MBC1 plays a role or anything like that.
It's a great question, and that definitely needs to be happening soon. We haven't looked at it yet, but that is the next step to do. We hope to, the greatest challenge is to measure transport with having the microelectrode as close as possible to that cell. So we have to try to be consistent with that, and when we're confident with that, we start measuring those components and see how they work together in that area. Thank you for your question.
So do you think there's any bicarbonate going through CFTR? So in your diagram, it's A2, working coordinately with CFTR.
I think from our latest publication, I think that A2 is the most contributor of the bicarbonate that is secreted. Of course, CFTR is also secreting bicarbonate, but in that paper, we showed that when we block CFTR, there is no bicarbonate or very minimal compared to the AE2. So the main contributor could be the AE2 that is secreting bicarbonate, and it is CFTR linked because when we inhibit the CFTR, the whole function of the cell shuts down.
Yeah, so is AE2, is that completely blocked by DIDs? Because our go-to is we add DIDs before we stimulate CFTR and then inhibit CFTR. So when we see an ion site dependent bicarbonate current.
For sure, one of the challenges is to find a better blocker that we know specifically would block that. As far as now, this is the best alternative we have. The idea is knowing that at least it blocks as much as any other blocker would. But of course, that's something that we need to look forward to try to see if we can find better.
Great.
Thank you so much.
Nice talk.
Thank you. Very nice.
Thank you, everyone.
Okay. Next speaker.
Hi, thank you for the introduction and also for the opportunity to share some of the work we're doing over at Boston Children's Hospital in the labs of Drs. Carla Kim and Dr. Ruby Wang, in trying to identify what pathways lead to ionocyte differentiation. To start, I have no conflicts of interest to disclose. And this conference has been especially great for anyone studying ionocytes since there's been so much that's been being discussed about them. And we've learned so much about how they may function in different contexts and different models, including the Engelhardt Labs ferret model, where they showed that ionocytes, when knocked out, there's a decrease in mucociliary transport and also in certain absorptives and fluid dynamics in the airway. And in our own lab, my colleague Stuart has also shown that when you knock out the ionocyte transcription factor, FOXI1, there's a decrease in CFTR-dependent function, and that is rescued by overexpression of FOXI1, which brings the ionocytes back. So clearly, we know these cells are important in some way or form, and we still need to learn more about that, but they might provide an avenue for alternative CF therapy. And that is why we still want to study this. But to even be able to study these cells properly, we know there are so few of them, and it's so difficult, right? So one thing we need to really understand is what causes these cells to be such few numbers, what regulates their differentiation and their numbers. And there are already some hints in the literature, and the speaker before me talked about Wnt signaling and LEF1, and that was awesome. I will be focusing on Notch signaling for a couple of reasons, including previous studies showing that when Notch signaling is inhibited in ALI cultures, HBEC ALI cultures, there's a reduction in FOXI1 expression, which also points to the fact that ionocyte numbers are reduced. Additionally, when the receptors when Notch receptors are blocked using antibody, you see a decrease in ionocyte numbers, as shown here. And not only this, but recent findings also showed a tuft ionocyte progenitor which expresses POE2F3 and ASCL2. And this links us back to Notch because ASCL2, this marker for the transcription, the ionocyte progenitor, has been shown to be regulated by Notch signaling. And it's kind of complicated, but then the summary is that Notch signaling could both upregulate and downregulate this ionocyte progenitor marker, ASCL2. Again, linking us to the fact that Notch signaling plays a role in ionocyte differentiation. And also, as was mentioned before, not in pulmonary ionocytes, but then in epidermal ionocytes, Notch inhibition increases ionocyte numbers, whereas Notch activation basically gets rid of ionocytes. And in the third panel, INJ, you do not see the FOXI1 stain in there. So overall, all this data tells us that Notch signaling is important, but it may be complex in how it's regulating ionocyte differentiation. And our lab also looked into this, and this is recently published in just last year. We tried to do the opposite and overexpress Notch in iPSC-derived airway cultures. And we did this using a DOX-inducible NICD-iPSC line. So upon administration of DOX, the intracellular signaling domain of Notch is overexpressed, and then we get activation of Notch. And we did this at 2 different time points. And what we see is that indeed we are activating Notch as seen by HEY2 expression. And we do see the expected changes in our secretory and ciliated cell populations, where secretory cells are going up and ciliated cells are going down. But we did not see any appreciable changes in FOXI1 expression. However, interestingly, CFTR expression at one time point of activation went up, whereas at the other time point, CFTR expression was going down. So this was an interesting result for us, and we wanted to find out a couple of things. It rose a couple of questions for us. One was, does Notch inhibition play a role in ionocyte differentiation from the epidermal ionocytes and also from the ASCL2 finding, and how Notch could both up and down regulate ASCL2? We're wondering, does inhibition play a role? And secondly, is there a temporal aspect to how Notch is regulating ionocyte differentiation? And that is from the earlier work that we did in our lab. So to test this, to first start looking into this, we wanted to do two things. One is inhibit Notch signaling using DAPT, as was done previously, but then do this at different time points, an early and a late time point in ALI differentiation. And then also we want to do activation of Notch signaling at an early and late time point using the same DOX-inducible NICD-iPSC cell line. And so to start off with the inhibition, what we did was we inhibited Notch signaling using DAPT at airlift. So when the cells were airlift at the beginning of the differentiation from basal cells, we started treating with DAPT. We did that for 3 weeks, and then after 3 weeks, we would harvest the cells and take a look at the cell populations. And then we also did a late inhibition where we allowed the cells to differentiate after airlift for about 3 weeks, and then we would start the treatment using the inhibitor. We would treat for an additional 3 weeks under the inhibitor, and then we would take a look at the cell populations. So just to start off with the early inhibition, and this is similar to what has been shown in the literature, that if you inhibit Notch signaling at the beginning of a differentiation, you see a reduction in ionocyte markers, so that's the punchline, right? BSND expression is down, FOXI1 expression is down, and as expected, the MUC5AC or goblet cell expression is down as well. To dive a little bit into this, Notch pathway is inhibited, as we can see. The secretory cell markers, club cells and goblet cells are both down, so we know our inhibition is working. When we look at ionocyte markers, we see the ionocyte progenitor FOXI1 expression is down. ASCL3 and BSND, the more mature ionocyte markers, are also reduced. In terms of ion channel, CFTR expression is way down, and we think this is because we're losing both our secretory cell population as well as our ionocytes. And additionally, we see a decrease in ATP6 VOD, which is another ionocyte marker, and also the potassium and sodium channels. And looking back at our TIP precursor marker, looking at POU2F3 and ASCL2, we see kind of a differential expression here, where POU2F3 expression is down, but ASCL2 expression is up. And we think the increase in ASCL2 is probably due to the fact that Notch signaling can both upregulate and downregulate ASCL2 expression. So in this case, we're blocking the inhibitor and then causing an increase in ASCL2 expression. On the other hand, when we do the inhibition at a later time point, so about 3 weeks after the culture has been allowed to differentiate, we see a reverse, right? So we see that ionocytes are not knocked out, but instead they might be increased as per the BSND and FOXI1 staining. And just to delve a little deeper again, the Notch pathway is truly inhibited as per HES1 and HEY1 expression. And our secretory cell markers are again reduced as expected. Mature, and we see that in this case, the FOXI1 ionocyte precursor marker is not reduced, but just maintained. However, the more mature ionocyte markers, ASCL3 and BSND, are increased. Additionally, the ion channel markers are down. CFTR is still down, but it's still at a higher proportion than what it was when it was knocked out earlier at the early time point. On the other hand, the ATP6 VOD, which is more specific to ionocytes, was increased. And again, we see that the precursor markers take the same pattern where POU2F3 is knocked down, but ASCL2 is up. So the inhibition is having a similar effect on the precursor, but it seems that the mature ionocytes are increasing. So the take home from the late inhibition, the take-home point is that the progenitor is still down. The ionocyte progenitor marker FOXI1 is unchanged, but then mature ionocyte markers are increased. And another finding that we saw at this stage, if ionocytes are maintained and secretory cells are knocked out, then what happens to the CFTR-dependent ion channel function? And when we did that experiment, what we saw was that it was basically unchanged at this later time point. Although we're losing all our secretory cells, our club cells and goblet cells, there's still CFTR-dependent activity that is similar to the untreated group, the vehicle group, the control group. So that was another interesting finding. And finally, we did the activation of notch signaling using the DOX-inducible NICD-IPSC line, where we did the activation after 3 weeks of differentiation, where the cells were a little bit more mature. And what we saw here was that BSND expression was basically not existent in our cultures. And this is preliminary. We're still working on this data and trying to get more information from it. But it points us to the fact that later on in the differentiation, notch activation might actually be inhibitory to the maturation of ionocytes. So putting it all together, this is the model that we are currently thinking of, that from the basal cell stage, notch activation might be required to reach a progenitor state. And that is why we have seen the results we have seen in the past and also in our own hands. However, later on from this progenitor state going into a more mature ionocyte state, you might need to inhibit notch signaling to drive the process forward. And the 4 big takeaways from this talk that I want you to have is: 1, it is true that early inhibition of notch signaling reduces ionocyte numbers, likely by inhibiting the progenitors. 2, that late inhibition of notch signaling still affects progenitors, but it increases the number of mature ionocytes, so it allows for ionocyte maturation. That 3, late activation of notch inhibits ionocytes development or differentiation. And then finally, in the mature airways or the more differentiated airways, the absence of secretory cells does not seem to, in this case, under how this experiment was done, affect the CFTR-dependent trans-epithelial function. And in the future, we still have a lot of questions to answer, right? Because this is very preliminary work. But we still need to look at the notch regulation to determine, is it doing this directly or is this doing this indirectly? Because within the time points that we're looking at, a time point of about 3 weeks, a lot could be happening. And it could be affecting another pathway that is having a more direct effect on ionocyte differentiation. So we have to figure out, is it direct or indirect? And then also, is it sufficient by itself, right? If it's not direct, what other signaling pathways may be involved and what combination of pathways potentially could yield the best results? And to do this, we are planning to test the sonic hedgehog, Wnt, notch, FGF, and BMP pathways using our newly generated FOXI1 fluorescent knock-in iPSC reporter cell line. And with this, we're hoping to do a kind of a semi-throughput test to see how various activators and inhibitors of these pathways affect ionocyte differentiation. With that, I'd like to say a very big thank you to my mentors, Dr. Carla Kim and Dr. Ruby Wang, for all their help in setting up this project and their guidance. I'd also like to thank Stuart for being there to help me through this process. I'm very new to the CF field, about a year old, and he's been very helpful with questions and then all the many questions I have. And also to my lab mates, Jenny and Alyssa, for the help with experiments, and to our collaborators, Dr. Thorsten, George Daly, and also Sharon Wu, who helped us to develop this new fluorescent line and also give us some reagents for some of the experiments we plan to do in the future. And I'll be happy to take questions.
Time for questions.
Great talk, Ian Thornell, University of Iowa. So I have two questions. So one is, have you looked at which Notch receptors or its ligands are involved in this interaction? And kind of along the same lines, have you considered that inhibiting gamma secretase might be doing something that's independent of Notch?
Yeah, so thank you for the question. Very great point. In terms of the Notch receptors, we haven't directly tested that. We have done some preliminary analysis looking at available single-cell seq data to see which populations are expressing the receptors and the ligands. And in terms of the progenitors that we have identified, the tip progenitor, I don't think we've seen a lot of those in the publicly available data sets, but some of the other basal cells express some Notch 1 or 2 ligands that we think, receptors that we think might be playing a role there, but we're not sure which one exactly is involved. And in terms of inhibiting gamma secretase, yes, it probably is doing a lot more than Notch. So that's one of the reasons why we're trying to shift away into a more direct approach, doing the NICD and then hopefully some other form of inhibition later on as well. Thank you.
Awesome, thank you. Looking forward to those future studies.
Thank you.
Yeah, you set us up because actually that was very similar to my question. I think when you set up the talking about what exactly regulates the frequency of this rare cell type in a heterogeneous epithelial population, I think that's where we're all like, oh yeah. Yeah, so Notch signaling, I mean, maybe this is a dumb question, but could you just like put a bunch of overexpress the ligands in different cell types with your new iPSC cell line? You can maybe overexpress it.
Overexpress
Specific ligands to look at what arm of the Notch signaling pathway, or and then you can play with timing and stuff like that. I was probably thinking about doing that.
Yes, so the ligand is an interesting question. One of our collaborators has this nanoparticle that has specifically the DLL4 ligand on it, and we've been trying to test that. We still have to figure out the concentrations, but that is definitely something that we can do. Thank you.
Yeah, the ligand question, I kind of had that one too, but I'll go in a different direction then. So with the relieval of Notch inhibition with your DAPT and the ALI, have you ever followed after a washout to see if they return at the same frequency or if they return at a different frequency?
At the moment, no. That is one of the experiments we plan to do, yes.
Very
But it would be interesting. And we're also thinking of doing activation and inhibition, trying to do different timings to see if that changes the population in any way. Thank you so much.
One last little quick one. Do you have plans to go into like the RBP-JK knockout mice, which are fully ciliated, that are the Notch deficient, to see if this holds in the animal model as well?
I think an animal model is something that we are thinking about. That would be a good one to try, actually. And then also even just overexpressing RBPJ with a viral construct is something that might be down the line as well.
Very nice talk. One trivial question. So have you tried, so you have shown multiple effects of inhibition, late or early inhibition or stimulation of Notch on the markers, various markers of ionocytes. You have shown a decrease in FOXI1 and the increase in BS and D. But there was a different thing that happened to CFTR. Have you tried to stain for CFTR and co-localize it with these cells and see which ones actually are expressing CFTR and which ones are not?
So CFTR staining, we just got the antibody. I've heard, I haven't done it actually, I've heard that it's very difficult and that the only really good antibody is in limited supply. So we are working on trying to do that. We have the antibody, we would see if we can do it in our hands. Right. Thank you so much.
Our last speaker is Sahira Hamad. She will talk about ELD 607 as a ORA1 antagonist.
Good morning, everyone. I'm Syra Ahmed, the Director of Research for LDEC Pharmaceuticals, and I'd like to thank the organizers at NACFC for giving us this opportunity to present our research on ELD 607 reverses goblet cell metaplasia in beta-ENaC mice. These are my disclosures. In normal airways, the airway surface liquid is properly hydrated. However, in CF airways, goblet cell metaplasia occurs, and this increase of goblet cells results in mucus hypersecretion, where you have dehydrated airways that results in impaired mucociliary clearance and also increases inflammation and resulting in airway remodeling. In addition to excess mucus production, in CF there is chronic airway infection, neutrophilic inflammation, along with lung damage, resulting in this CF vicious cycle, where each of these symptoms are there and also enhance each other to ultimately result in increased inflammation and lung remodeling. And a key component that regulates inflammation is this ORI1 calcium channel that is found on the membrane of many cells. ORI1 allows for calcium influx to occur, in which elevated calcium intracellularly results in pro-inflammatory responses. So calcium entry drives adaptive immune responses, and therefore ORI1 regulates calcium-mediated inflammation and airway remodeling. And this occurs in the following method. When the stimulus is present, you have a series of cascading events that occurs, resulting in calcium being depleted from the ER. This in turn causes STEM1 to activate and translocate, in which it binds to ORI1 calcium channel, activating this calcium channel, opening it, and allowing for calcium to enter the cell, that then activates NFAT to translocate into the nucleus, that then activates transcription factors for pro-inflammatory responses, and also results in secretion of cytokines, mucins, and proteases, ultimately resulting in inflammation and airway remodeling within the airways. And it is necessary for STEM1 to bind to ORI1 for this activation of ORI1 to allow for calcium influx to occur. So if STEM1 is bound to ORI1, you have influx of calcium resulting in inflammatory responses. So we took lungs from normal and CF patients and measured for this co-localization of STEM1 and ORI1 within these lung tissues. And we saw within the epithelia and immune cells of CF, there is an increase of this STEM1 ORI1 co-localization, where CF disease is present. However, we did not see co-localization of STEM1 and ORI1 in the alveola, where CF disease is not present. Therefore, this suggests that ORI1 is hyperactive in CF lung, and not in the data that I'm showing today. We have also obtained CF lungs to look for ORI1 expression, and we saw that there's an elevated expression of ORI1 within CF lungs, despite these lungs obtained from patients receiving ETI, suggesting that inflammation still persists. So we developed ELD 607, which is an ORI1 modulator. ELD 607 binds to ORI1, preventing calcium influx from occurring. This leads to ORI1 internalization, and ORI1 is also sent to lysosome for degradation. So with less ORI1 present on the membrane, you have less calcium influx occurring, and this ultimately reduces inflammatory responses to reverse lung damage and reverse lung remodeling. ELD 607 is an 11 amino acid peptide, and it's highly specific for ORI1, so it does not bind to ORI2 or ORI3. And when we screen it for its safety, we saw that there was no cellular toxicity in vitro, and also did not have any off-target binding when screened against a PanLab 44 target screen. And more specifically, it did not have any effect on hERG. And when we administer ELD 607 by inhalation, we saw that it remains mainly in the respiratory tract. And more specifically, it retains activity in highly proteolytic CF sputum where protein and peptides are normally degraded. So to test ELD 607 within a CF system, we used a murine mouse model that overexpresses beta-ENaC, in which as ENaC is overexpressed, as you see in these airways, this results in dehydration of the airway surface liquid that results in goblet cell metaplasia and mucus plugging. So we used a modified model based off of Marcus Mall's paper from 2008 that showed that these beta-ENaC mice develop spontaneous inflammation by day 14. So these mice, one day after birth, were dosed daily with ELD-607 through intranasal instillation for 10 days straight. And they were observed, and then 24 hours after the last dose on day 10, these mice were harvested on day 11. But while we were observing these mice, we noticed that those beta-ENaC mice that received ELD-607 had improved survival compared to the non-treated mice, showing that ELD-607 is improving their health. But to determine if ELD-607 is targeting ENaC to improve their overall well-being, we isolated trachea from these mice and measured for short current circuit in oozing chambers. And when amiloride was added, there was no change in ENaC activity to show that ELD-607 is not targeting ENaC to improve the overall well-being of these mice, but it is reducing or improving their health. So we went back to the airways and collected bronchoalveolar lavage from these mice. And if you look in the bottom left panel, beta-ENaC mice have increased neutrophilia within their airways. However, after treatment with ELD-607 for 10 days, there was a significant reduction of neutrophilia, as seen in the bottom right panel. And the leukocyte levels were normalized to what we see in the wild-type mice. Furthermore, we looked at biomarkers of lung damage that include neutrophil elastase, total protein, and lactate dehydrogenase that are elevated in our beta-ENaC mice. However, after treatment with ELD-607, we saw a reduction of these pro-inflammatory biomarkers for lung damage, and they were also reduced to levels that we see in wild-type mice as well. And we further looked into pro-inflammatory cytokines that include IL-6, TNF-alpha, and Kc that are elevated within the beta-ENaC mice. And we saw similar results as well, where they were significantly reduced and in the mice that were treated with ELD-607 and restored to baseline levels that were seen in wild-type mice as well. We next took lungs for histology, initially stained them with H&E to look at airway alveolar thickness to look for inflammation. And if you see in the bottom left panel, beta-ENaC mice have increased airway thickness, leading to increased inflammation. However, after treatment with ELD-607, there was significant reduction of this airway alveolar thickness. And these lungs were normalized to levels that you see in the wild-type mice as well. We next perform AB pass staining to look at goblet cells within the airways. And if you see in the bottom left panel, there is goblet cell metaplasia occurring within the airways of these beta-ENaC mice. And after treatment with ELD-607, we also saw a significant reduction of goblet cells within the airways. And they returned to levels similar to what we also see in the wild-type mice as well. And as goblet cells leads to mucus hypersecretion, to increase mucus plugging within the airways that you see in the left panel, we also saw a significant reduction of mucus plugging within the airways after treatment with ELD-607. So these results were promising, and then when we compared the results that we saw from these studies along with our other animal models that include mice infections, along with our ferret studies in conjunction with John Inglehart's lab, we saw similar effects in our endpoints that are seen in clinics with Trikafta, in which ELD-607 in our animal models decreased neutrophilia and inflammation, also decreased lung bacterial counts, and decreased mucus, inflammatory cytokines, and decreased airway wall thickening. So if I revisit back to this initial CF vicious cycle that I mentioned at the beginning of my talk, ELD-607 has shown to reduce airway infection and reduce neutrophilic inflammation. It also normalizes mucus production and reverses lung damage, ultimately providing positive evidence that ELD-607 can help reverse lung remodeling. And with this, I'd like to thank our LDEC team, along with the Robert Tarrant Lab at Kansas, Dr. Barbara Grubb and Troy Rogers at UNC for their help with the oozing studies, and the wonderful cores at UNC for the help with this research. I'd like to thank you all for listening today, and I'd be happy to take questions.
Hi, Victoria Momeyer from the CFFT lab. Really nice data. I was personally just curious, did you do any sort of analysis on the different cell types of the airway to see if the distribution was impacted in mice that were treated with the ELD 607 versus wild type?
That's a very interesting question. We actually have not done that, but that would be great to look into.
Thank you.
Yes.
Hey, very nice talk. So my question for you is, I like the model that you're using and the original mouse model, beta-neck mouse models, that the disease starts very early, so you started the treatment with the drug very early. My question is, with the mice that they survive, if you do the washout of the drugs, do they reestablish the disease?
Yes, so we have not gone further into it, but that's something that we could look into. I do know for these beta-ENaC mice that if they go on longer, eventually, because they are designed to survive, the inflammation does eventually start to go down. So that's why we kept it to within the first 10 days.
So here, very nice. Since the ELD does not affect ENaC, have you tried combining it with ENaC inhibitor like siRNA?
We have not done that yet. One of our plans is to do parallel studies with ENaC inhibitors along with other drugs too, especially ETI.
Right.
Excellent talk. My question is, my name is Nam Stewart at Stanford. The question is, your compound is peptide, you said, 11 amino acid. Then it may be degraded if it facing to like neutrophic elastase or all those inflamed human airway.
Yes, so we actually have tested it in CF sputum and also specifically incubated the peptide with neutrophil elastase, and we saw that it maintains stability even up
So it's
to...
resistant to...
Yes, yeah, we've designed it so it does not degrade from neutrophil or other serine proteases too.
So you're meant to aerosolize it, not give it systemically, right?
Correct, yeah. Our plan is to use it as an inhaled therapeutic.
I just would like to thank all speakers.